Hello, everyone. This is a 6-part course, Cancer Prevention through Methylation and Oxidative Balance. In this course, I'll be exploring how methylation and oxidative balance work together, and they work together mainly to regulate gene expression, regulate DNA repair to prevent cancer. This course is broken down into 6 parts, and each part will have its own video. So welcome to video 1, part 1, and let's dive right into it. First things first, starting from food. When we eat food in the form of carbohydrates, fats, or proteins, our body breaks them down into small nutrients. These small nutrients are in the form of glucose, amino acids, or fatty acids. Now, the mitochondria that live inside almost every cell in our body use these nutrients to generate ATP or energy. This energy that the mitochondria produces is generated via a process called oxidation. In doing so, in producing energy, the mitochondria also produces what's called ROS, or reactive oxygen species, as a byproduct. Now, these byproducts are in the form of reactive oxygen species, or ROSs, are a double-edged sword. And I'm saying a double-edged sword because they can work both ways. They can act as a defensive mean to our cells and protective mean, and they can also damage cells and DNA if we have too much of them and we don't have enough antioxidants to neutralize them and keep them in check. And if that's the case, this leads to oxidative distress, oxidative imbalance, oxidative overload. All of these are the same terms and they mean the same thing and they're used interchangeably. If this oxidative overload persists over time, it ends up damaging cells and damaging DNA. Now, our body is equipped to always bring us back to balance by producing antioxidants and by means of detoxification. Now, detoxification means how the body automatically cleans itself from waste, from toxins, and from chemicals that we are exposed to every day. When detoxification is overwhelmed or fails, this can lead to cell damage and distress. So to summarize before we leave this slide, oxidative overload or oxidative distress can develop in one of 2 ways. 1, not enough antioxidants to fight the burden of excessive ROSs or oxidants. 2, poor detoxification. And this combo of poor detoxification and not enough antioxidants can create an environment that promotes DNA damage, and ultimately it can increase the risk of cancer. So let's go to slide 2, which is the most important slide of this discussion here. First things first, to list out some of the factors that increase the quantity of ROSs in the body. 1, processed foods and additives, alcohol, added sugar, a lack of physical activity or exercise, chronic perceived stress. So all of these poor lifestyle habits can cause excessive amounts of reactive oxygen species and oxidants to build up. So let's start from the left of this slide. When ROSs or oxidants level exceed the body's antioxidant capacity and detoxification is insufficient, DNA damage occurs. This DNA damage triggers chronic inflammation, and over time, the function of our mitochondria that produces energy from food starts failing. And the next thing that fails is what's called apoptosis. And failing apoptosis means the body is losing its ability to remove old, damaged, or abnormal cells. And as a result of this failure in apoptosis, damaged cells continue to survive and replicate uncontrollably. This uncontrolled chaos leads to tumor formation and what is called metastasis. And metastasis simply means the spread of cancer. So in the next slide, what I have here is a lab marker that we can use and we can screen for as part of our yearly screening physical. And we can get this done via multiple companies that provide this without any doctor's prescription. And this definitely should be part of our yearly screening. And it really gives us an idea on how our mitochondria and how our DNA is doing from the inside. And these are my references. And that's about it, folks. Thank you so much and have a good day.